Welcome back, students, to the class of histology. So, in the series of histology of alimentary canal, this is the third lecture, and it's Priya Ambekar, assistant professor in zoology, KANMS Arts, Commerce and Science College, Satana, District Nasik, Maharashtra. So today in this lecture we are going to discuss about the histology of small intestine. As all of you know that small intestine is around 6 meters long, and first part of the small intestine, that is duodenum, second part is jejunum, and third is ileum. So today we are going to discuss about the first part, that is called as duodenum. So around 7 meters long, it is the length of small intestine. It is divided into the this duodenum, jejunum, and the ileum. So, as all of you know, that whatever food is received in the small intestine from the stomach, it is now mixed with the HCl and various enzymes of digestion, and that is called as chyme. And this chyme will enter into the duodenum part, which is around 25 centimeters long, and the small intestine receives the digestive enzymes from the pancreas and from the bile. As all of you know that pancreas, it is the mixed gland that is having the exocrine as well as endocrine function. So exocrine part of the pancreas secretes the pancreatic juice, which is the mixture of the all the digestive enzymes, which for example there are lipases for the lipid digestion, there are proteases for the protein digestion, there will be nucleases for the nucleic acid digestion, and so on. And it receives the bile from the gallbladder. As all of you know that liver synthesizes the bile salts are, they are helpful in the digestion of lipids. They break down the larger particle of lipids into the small micelles. So this everything is received by the duodenum, and it will assist in the process of digestion, and simultaneously there will be the absorption of the nutrients, digested nutrients that occur in the duodenum. So, as we have discussed earlier, also that there are four layers of the this linings of the gastrointestinal tract. The innermost, that is mucosa, followed by the submucosa. External to the submucosa will be the muscularis externa, and finally, last one, outermost, that is serosa. And specialty of the linings is in the mucosa lining. Mucosa lining is further composed of the epithelium, followed by the lamina propria and muscularis mucosa. So the epithelium here, as you can see, there are the folds, and these folds are called as villi. So finger like projections of the folds of the epithelium are called as villi, and epithelium plus lamina propria plus muscularis mucosa, all of them will be present in these folds, and these folds are called as villi. And as all of you know that intestine is not only the part of digestion, but it is the site where absorption of the digested material will also takes place. So इतनी मेहनत करके हम लोग जो खाना पचा रहे हैं उसमें से कोई भी मोनोमर्स यानी कि डाइजेस्टेड फूड पार्टिकल जो है वो वेस्ट नहीं जाना चाहिए. इट शुड बी एब्जॉर्ब completely within the intestine. So that's why to increase the area of absorption, there is a folding. So म्यूकोसा की जो फोल्डिंग्स है जिसको हम विलाइज बोलते हैं, these are only to increase the area of absorption. So as much possible, that much absorption of the food material should be there. So that's why villi are present. These are the finger like projections, and along these with this villi, the outermost layer, that is the epithelial layer of this intestine, is made up of columnar cells, which are column like cells. And these column like cells will have the brush border, and they will show the microvilli also. So these microvilli and villi will increase the area of absorption. Here one thing you can notice that the depth of this the deeper side of these microvilli are called as crypts of libercules. These are the depressions of the this mucous lining. Then there is a presence of specialized goblet cells which are present in the crypts of the libercules. And this is a simple columnar epithelial, or it is having the brush border, so villi are present. Then we will discuss about it in detail. So you can see here that the finger like projections or extensions of the lamina propria and the epithelial lining are called as villi, and they are covered by the simple cuboidal epithelium, which enlarges the luminal surface by 10 folds, which are called as villi. And these villi having the different length in the different part of the small intestine. It is around 1.5 meter tall, millimeters tall in duodenum, 1 millimeter tall in jejunum, and smaller but a little bit having lesser length in the ileum. So and further, the epithelial lining cells will also have the small finger like projections, which are called as microvilli, which will increase the area by 20 folds, and the spaces between the two villi, that depression, it is called as crypt of Libercunes. So as you can see here, these are the villi which are finger-like projections, and the spaces, depressions between the two adjacent this villi, it is called as crypt of Libercunes, and there are the columnar cells which will have the brush border. So this is a specialty of the mucosa layer of the duodenum. So you can see that mucosa layer is consisting of the three layers. This slide is very important. The mucosa lining is made up of the simple columnar epithelium, then which is followed by the lamina propria and then muscularis mucosa. So the lining of the duodenum, it is nothing but the columnar epithelium having the brush border to increase the area of absorption. There are the goblet cells, specialized cells, which are present in the deeper crypts of Libercunes, which secretes the mucin. This mucin, when released into the lumen of the duodenum, it will get mixed with the water and it will form a slippery substance that is called as mucus, which is helpful in the this movement of the food without any friction, because the food has to travel the long 7 centimeters long tube. So without any friction it should be passed, so the mucus will get make it slippery. The surface will be made slippery by the mucus, so that food can be easily passed. Food means the chyme can be easily passed through that lining. Then there are specialized M cells. M cells are the phagocytic cells. If there is any kind of antigen which is present in your food, that will be identified and captured by the M cells, and that will be presented to the antigen processing cells which are present in the lamina propria. So M cells are also important. The main part, that is crypts of Libercunes, which are the depressions. The lamina propria of the mucus is the loose connective tissue that is rich in the lymphoid capillaries, and it is having the core of the villus, and it presents the having the lacticles. The crypt of Libercunes, which are very important, they are present deeper in the lamina propria between the base of the villi, and they are richly vascularized. They have the rich supply of the blood vessels, and they form a glandular structure that is nothing but the crypt of Libercunes. And these gland forms the intervillous space of the muscularis mucosa. And there are specialized cells, that is regenerative cells, which will continue to proliferate to form the new cells. There are the Paneth cells. These Paneth cells are important one because they will secrete the lysozymes, defensins, and antimicrobial agents and tumor necrosis factors. So lysozymes will kill the entering bacteria which will enter into your body along with the food. So to kill those bacteria, there are the lysozymes which are secreted by the Paneth cells. Then after that, there is a layer of muscularis mucosa, which is composed of the inner circular and outer longitudinal muscles. So as you can see here completely, that is, there are the two villi, neighboring villi are there, and in between the gap of the two villi there is a crypt glandular structure, that depression that forms one gland-like structure, that is called crypt of Libercunes. And these are having the specialized cells. There are surface absorptive cells having the brush border. You can see here clearly the finger-like microvilli are present, that is called as brush border columnar epithelial layer. And there are goblet cells which will secrete the mucin, enteroendocrine cells which will secrete the various hormones such as gastrin hormones, which will help in the process of digestion. There are regenerative cells which will replace the old and damaged cells. There are Paneth cells which will secrete the lysozymes, tumor necrotic factors, and so on. So these are the very important, this is a very important slide. Then next to the mucosal there is submucosal which is nothing but the dense collagen connective tissue. It is followed by the muscularis externa made up of circular and longitudinal muscles, and followed by the serosa. All these layers, that is muscularis externa and serosa, will help in the process of compression and relaxation of the alimentary canal. As the food passes through the alimentary canal, the passage of the food will be this what it will give the strength to the alimentary canal tube. So स्मॉल इंटेस्टाईनमध्ये जी पण फूडची मुवमेंट होते, ती मुवमेंटच्या वेळी रिलॅक्सेशन आणि कॉन्ट्रॅक्शन जे होतं इंटेस्टाईनचं, ते त्याला स्ट्रेंथ देण्याचं काम या मस्क्युलॅरिस एक्स्टर्ना अँड सेरोसा लेयर्स करत असतात. सो थँक्यू व्हेरी मच. We will meet in the next lecture and we will discuss about the histology of the rectum.